I just showed you just a minute ago the middle meningeal arteries coming directly, from the basically the central circulation. So one of the things that can very rapidly happen if we have a fracture here in the skull is we can develop a hematoma. And this hematoma then is in that tight space between the skull and the dura. And so again, it's a closed volume system. You pump a lot of blood in. What's going to happen is you're going to compress the very compliant brain. And so what happens is the brain then shifts and you get what's called tonsillar herniation. This is called the uncus of the temporal lobe. And it herniates and it presses on the pons. And when it presses on the pons, that's where our respiratory center is and you quit breathing. And so somebody that develops an epidural hematoma, usually the result of a skull fracture. Somebody who has an epidural hematoma. This is a true medical emergency. Those of you who are trained, you probably study this. If you had advanced CPR, if you come across a patient that has rapid change in consciousness, obviously there's a fracture. You can palpate the fracture. This patient's going to die if you don't take something, an ice pick, screwdriver, something, and just stick it through the scalp and allow that blood to be evacuated. Otherwise, the uncal herniation is going to end up causing the patient to expire. Yes?
So you didn't break the skull and let the blood flow out?
Yeah, you just let the blood come out.
So you have to penetrate the skull then?
Yeah, well, there's not, first off, the middle meningeal artery is not the aorta. You're not going to die in 10 minutes just letting the blood come out. You're going to hope that that's going to clot, okay? But if you don't decompress, the brain now is herniated against the brainstem, and you're going to quit breathing. You basically are already unconscious. So again, this is a patient that you come across that has rapid loss of consciousness in the setting of a skull fracture. You have to evacuate an epidural hematoma very quickly, or else that patient will expire. Okay, any question about, this is called an epidural hematoma. The source of the blood from an epidural hematoma is what? Middle meningeal artery, right. That's correct. It turns out that there is another kind of hematoma, which is called a subdural hematoma. The subdural hematoma accumulates in this space right here. So the subdural hematoma would accumulate in this space, okay, between the dura and the brain. It's typically caused by a rupture of the small penetrating veins, okay? So it's important to understand, I know we haven't looked at it yet, but the blood vessels for your cortex are all on the surface. They're all outside. They're all on the surface. And so you've got all these little veins on the surface of your skull. They're underneath the dura. So this is a subdural. You break those, subdural hematoma. And then the setting for a subdural hematoma, unlike the epidural hematoma, is something that is really kind of insignificant. You bump your head on a shelf, okay, or you, the classic example that we used to give was you're out hanging up clothes on a clothesline. I bet y'all have never hung up clothes on a clothesline. Y'all know what a clothesline is? And so you're out hanging up clothes on a clothesline, and those posts, you bump your head on the clothesline, the post of the clothesline. Something that's really not really, really bad. You haven't had a fracture, you haven't had anything terrible happen, but you end up, after maybe a week or two, you end up with a headache. You've probably already forgotten that you even hit your head. And so you develop all the same symptoms as an epidural hematoma. You end up with a dilated pupil on the same side. You end up with, usually there's some degree of confusion, some cognitive function that's not quite right. And typically you get a contralateral hemiparesis. So basically you kind of get a numbness or tingling in your arms and legs on the same side. So dilated pupil hemiparesis, some changes in consciousness, along with the headache that's developed slowly over a couple of weeks. and you probably have a subdural hematoma. Both of these are treated the same way. They have to be evacuated surgically. These are surgical problems. If you don't fix both of them, you'll end up dying. They'll produce, either one of them will kill you. The epidural is a true emergency, has to be done typically in the field. The subdural, you wait. Many times I've been on call for the hospital and diagnosed an epidural hematoma, and you don't have to rush. You just call up the neurosurgeon and say, hey, listen, I got somebody here with a subdural hematoma. You want me to go ahead and put him on the schedule for tomorrow? Okay, so it's not the urgency that you have with the epidural hematoma. And the reason is because this is a venous bleed, and the epidural hematoma is an arterial bleed. Okay, so now you know about subdural and epidural hematomas.
Did you say the contralateral increases was tingling on the same side of the damage, or is it on the other side?
It's always on the other side. Contralateral means opposite side.
Yeah.
The reason is because the tracts are crossed.
But like by contralateral, does it mean like literally on the other side of the body?
Yes.
Side, okay.
Contralateral means other side. I'm sorry, I didn't maybe. You'll hear these words. You'll understand what they mean as we're doing a lot. But anyway, ipsilateral, same side. Contralateral is opposite side. Okay. So, our brain, any questions so far? Okay, isn't this fun? Now, we're going to talk about the cerebrospinal fluid. This is also really cool. We have another circulation inside of our brain, which is the production and reabsorption of cerebrospinal fluid. Okay? And it turns out that cerebrospinal fluid is primarily produced in the choroidal plexus of the lateral ventricles. So it's kind of produced in the lateral ventricles. It then goes to the foramen of Monroe, which is right here, into the third ventricle. Then it comes down the aqueduct of Sylvius into the fourth ventricle. And then it exits through the foramen of Magendie and Luschka. And then it comes back up. And it comes back up and it's reabsorbed in the arachnoid granulations, okay, of the superior sagittal sinus. So now I'm just giving you the circulation. That's all you need to know. Okay. Choroidal plexus, foramen of Monroe, third ventricle, aqueduct of Sylvius, foramen of Magendie and Luschka, around through the subarachnoid space. So the fluid percolates down, comes back up, and all the cerebrospinal fluid, or almost all of it, gets reabsorbed in the superior sagittal sinus, which is that big vein which is right here, right in the middle of your head. Okay? It's reabsorbed in the arachnoid granulations of the superior sagittal sinus. Okay? But you got to come up, as a medical student, come up with all these little rhymes and riddles. It's the thigh bone connected to the, leg bone. And so you have to come up with a little mnemonic, that kind of helps you remember. But anyway, the choroidal plexus is in the lateral ventricles, okay? And that cerebrospinal fluid, this clear, beautiful fluid, comes out through the lateral ventricles, comes through the foramen of Monroe into the third ventricle. Third ventricle then drains into the aqueduct of Sylvius. Aqueduct of Sylvius then drains into the fourth ventricle. It then exits through the foramen of Luschka and Magendie, comes up through the subarachnoid space to the arachnoid granulations, which are in the superior sagittal sinus. And that's the way cerebrospinal fluid percolates inside our brain. That's how we float our brain in cerebrospinal fluid. Okay? We'll talk about it some more. You'll learn all the names. It just takes a little time to do that. Now, the reason that this is important, is because there are some very serious problems that can happen with this circulation. Okay, the very first problem I want to talk to you about is a problem where we obstruct the obvious place here is the aqueduct of Sylvius. So actually, this is actually probably a little better diagram to understand the proportions of the anatomy here. So anyway, the fluid is being produced here in the lateral ventricles, okay, comes into the third ventricle here. The third ventricle, then it has to go through this little tiny tube, which is called the aqueductus sylvius. And then it gets down into the fourth ventricle here, and then it kind of exits, and then kind of percolates back up, and it gets absorbed up here like that. So kind of that's, again, that circulation. So the reason this is important is because this is a very tiny little tube, okay? And we produce a lot of cerebrospinal fluid. And so anytime you're producing a lot, and it has to go through a little tiny tube, you can imagine that if you have some kind of a stenosis or an obstruction, then that fluid is not going to be able to get out. It's going to be stuck. And so it turns out that about 1 in every 500 bursts, that's a lot, 1 in every 500 has an internal obstructive hydrocephalus. Okay. That's a lot. That's a lot of people. Now I bet somebody in here has known somebody that has had or is familiar with this word hydrocephalus. Anybody? Yes, in the back.
That's water in the brain, right?
Right. So do you have somebody, you know somebody that,
No, but I never knew her. My mom, she passed away.
okay? So most of this happens, the setting that we see the internal obstructive hydrocephalus is mostly in young infants, young kids. And, yes, question?
How do you fix this?
Well, we're going to talk about that in a minute. Let's diagnose it first. Okay? And so, how many of y'all have children? Anybody here have children? Have you ever been with someone to a pediatrician? Y'all still go to pediatricians? Have y'all graduated from pediatrician? Okay. All right. Well, when your mom used to take you to the pediatrician, you have no clue because you won't remember this, but when you went to the pediatrician, when you were newborn, all the way up through newborn, through toddler, through kind of infancy, through toddler, up in that kind of less than 2 years old kind of range. One of the things that the pediatrician does is he takes a tape measure and he wraps it around your head. And he just measures your brain circumference. And we have these tables that are normograms that say a normal person should have a head circumference of this or that. And if you're into the highest 3 percentile, then you are suspect for having this problem, hydrocephalus. So the infant, remember the bones aren't fused. So in a baby, the bones just expand and your head gets bigger. And so you get a big head. You get a really, really much bigger head. And it's really not high tech. All the pediatrician does is take a tape measure and wraps it around the baby's head. And if you're outside the nomogram, then you have to go get a MRI scan and see if you've got this problem. Okay? Really not terribly high tech. That's how you diagnose an internal obstructive hydrocephalus. Just as an antidote, a few years ago I had a student actually in another class. It wasn't this class, but it was a graduate class I was teaching. After the class, we'd been talking about this and how we treat it, and he came up and said, Dr. Reitlinger said, I was born with that and I have a shunt. Sure enough, he had a shunt. So the bottom line is, if you don't treat it, you'll become mentally retarded and eventually die at a very early age. But if you treat this problem, you can be absolutely normal, just like the graduate student in my class. He was bright, otherwise 100% normal. And so the question is, how do you treat it? You do a shunt. Yeah, you do a shunt. So where do you shunt? Shunt from where to where? You got to go from where the fluid's being made. So typically, it's really actually quite easy to do because if you have hydrocephalus, the brain has already kind of been thinned out. So all you have to do is take a needle and just kind of randomly stick it into the lateral ventricle, come up to what's called an Ommaya reservoir, which is a little pump that's connected to a tube, and we drain the fluid either into the right part of the heart, into the right atrium of the heart, or we drain it into the peritoneal cavity. That's called it's called a ventriculo-atrial or ventriculo-peritoneal shunt. What it does is it provides another pathway for the fluid to get from the lateral ventricle where it's being produced into where it can be reabsorbed in the heart or in the peritoneal cavity. Yes?
So is that like a permanent thing? Like the shot will always be there?
Yes, you have to have it the rest of your life. As an infant, if you go over to the Dell Medical, the Children's Hospital, if you look at the OR board, you can almost be guaranteed every day you'll see one or two cases which are called shunt revisions. Okay, so what happens is when you're a baby and you get this done, you grow and you grow and grow. You have to actually lengthen the tube. You have to make it longer and longer. When you get to be an adult, you really don't have to. Yes?
Are there any restrictions to people that have a shunt, like can they do sports or?
Yeah, I imagine they need to be a little careful. Yeah, because they've basically got this valve that's underneath the skull. So I imagine they need to be a little bit careful, but they live a normal life. They play tennis, probably wouldn't want to go boxing or, I don't want to play, football for protein or something like that. But, so anyway, that's called internal obstructive hydrocephalus. Now there's another kind of hydrocephalus which occurs, and that is if the arachnoid granulations, so this is a blockage. And by the way, in this class, we're not going to study all of the different things that can happen here, but the obstruction of the aqueduct is the most common way that you get an internal hydrocephalus. It's congenital, you're born with an atresia or stenosis of the aqueduct, and then that causes the fluid to back up. There are several other things. One's called the Arnold-Chiari malformation, which obstructs the foramen Lusca-Magendie. That is another common problem, which can be a developmental problem. And basically, you can obstruct anywhere, but it's just more common in that aqueduct area. Was there a question?
Yeah, you were saying the fluid drains to the heart. Why does the fluid drain to the heart quicker?
Well, because fluid always goes downhill, right? So you have higher pressure in your head than you have in the right atrium of your heart. What's the pressure in the right atrium of your heart? And it's about 4 or 5 at the most millimeters of mercury. The pressure in your head is 10. So you're going to get fluid flow through the valve. Yeah.
So are you draining cerebrospinal fluid?
Yes.
Okay.
Yes.
So why would you put that into your circulatory system? Is it just harmlessly absorbed?
Oh, absolutely.
No, okay.
That makes no difference. That just gets absorbed into your circulation. When you put it in the peritoneal cavity, it gets absorbed through the peritoneum. Okay. So anyway, what I was going to say is that there's another kind of hydrocephalus, which is a non-obstructive. If you can get a non-obstructive hydrocephalus if the arachnoid granulations don't absorb the fluid, and that can happen after you have meningitis. And so people that have meningitis can get a form of hydrocephalus because the arachnoid granulations have been damaged by the meningitis. There is another form which, the last I heard, there were 500,000 Americans, 500,000 older Americans have another problem called low pressure hydrocephalus. And this is a problem where there's really not an elevation in the pressure, but what happens is you get spikes. And so instead of the pressure being relatively constant, you get these wild swings in pressure inside your head, and that causes dementia. And so a lot of people actually get diagnosed with Alzheimer's disease actually have low pressure hydrocephalus, and they can actually be cured by doing a shunt. So how many people are sitting around in nursing homes just because they haven't had a good screen, they haven't been actually evaluated for this low pressure hydrocephalus? It's a travesty. This is a terrible problem that we don't address here in our own country. We brag about being the best in the world, but we fail to take care of our own. And so, as I said, we make a lot of cerebrospinal fluid. We make 600 milliliters a day. Over half a liter a day of cerebrospinal fluid, half a quart. That's how much your brain is producing. And so the total volume, if you look at the total volume in the subarachnoid space and in the cisterna, you only have 140 milliliters, so basically it turns over 3 or 4 times a day. So it's continuously flowing. And so the other purpose, cerebrospinal fluid is it floats the brain, it gives us mechanical cushion, but it also provides glucose to the ependymal cells in the ventricle. So it's actually providing nutrition to the brain. It typically is very low in protein, and it should have no red blood cells, and at the most 1 or 2 white blood cells in a milliliter sample of the cerebrospinal. It's a clear, colorless fluid. You sample it by doing a spinal tap. Now, there are a number of reasons where we diagnostically look at cerebrospinal fluid. Okay, can you give me an example of why we would do a lumbar puncture and why we would look for something bad in the cerebrospinal fluid? Yeah.
It's for arachnoid damage.
Okay. Give me an example.
So for meningitis, the non-obstructive hydrocephalus.
Okay, the non-obstructive. So if you have meningitis, and I do a sample on cerebrospinal fluid, what is the cerebrospinal fluid going to look like?
It might have some proteins in it.
It's going to have inflammatory proteins. What else?
Maybe some proteins from damage. It's just damaged cell parts from the arachnoid.
Okay, it's going to have some cells, and so if you have an infection, what kind of cells are you going to see? You're going to see white blood cells, right? So you're going to see a whole bunch. You're going to see 10,000 white blood cells. You're going to have a bunch of protein that's going to be up, and the glucose is going to go down because the infection, if you have bacterial meningitis, is going to be burning up glucose. Okay. And remember, I told you that one of the other things that can happen is if you hit your head real hard, you can fracture the cribriform plate. And so here you have a patient that comes into the emergency room. And so I have a patient that comes in, and the patient says, "Doctor," says, "I just have this continuous fluid coming out of my nose." And you're the emergency room doctor. What are you going to do? Surgery? There's an old saying, a chance to cut is a chance to cure. You just want to know. No, diagnostically, let's be smart. What are we going to do? We got somebody with something coming out of their nose. Okay, it can either be cerebrospinal fluid or it's snot. Okay? So we're smart. We're biomedical engineers. We're going to do an assay on that fluid. What are we going to look for? What are we going to look for? You're the emergency room doctor. Okay, so if it's snot, it's going to have a whole bunch of protein, right? Next time you go out and feed boogers, it's going to have green protein. It's going to be rich in protein, right? Okay. And cerebrospinal fluid shouldn't have hardly any, right? Okay, so that's the way. If you're the emergency room doctor, you're just going to collect a little sample. You're going to send it off to the lab. You're going to do an assay, and then you can tell if you've got a cerebrospinal fluid leak. If you have a CSF leak, you got to go in and repair it. You're right. You want to be a neurosurgeon, right? You want to go in and you got to repair it. So you got to repair the dural tear. You got to close off the opening. Why? Because the nose is a terribly dirty place. If you've got a leak with cerebrospinal fluid coming out, guess what? The bacteria can go the other way, right? So you're going to end up with a terrible infection if you're not careful. So cerebrospinal fluid can tell us a lot. We can also tell if people have MS and other diseases by doing samples of, just checking the cereal spine fluid. So are there any questions about maybe what we've discussed?